In our last segment, we talked about the fact that life is due to proteins providing a structure, and then those proteins responding to signals which cause them to move and change shape, and the movement is what produces the characteristics of life. Well, there's a very important insight about this. Proteins have to have a regulated environment. You can't have a pool of proteins and have an organized life out of it for a very simple reason. Proteins have to be at the right pH, acid or base. They have to be in the right salt balance. They have to be protected from signals that shouldn't be connected to them. They have to be more or less isolated from the pond in their own environment. So a skin, for example, isolates me as a human from the environment on the outside, so that inside my body I can control the pH, the temperature, the salt balance, these elements that are important for the function of proteins. If the proteins, as I said, were just out in a sea, they wouldn't be able to function because you need to control the environment. Well, how do you control the environment? The answer is, well, in a human, we put skin around us. But in fact, when we look at the cell, there's the equivalent of a skin around the cell. The skin is called a membrane. All cells, all living things on this planet have the same basic structure of a membrane. It's so fundamental to life that every organism on this planet, the cells of that organism have the same basic structure. A membrane is an assembly of molecules called phospholipids. Phospholipids are molecules that have a polar positive or negative charge on one side, and the lipids are nonpolar. Lipids are like oil. The phosphate groups are polar like water. Remember, oil and water don't mix. So I have a molecule called a phospholipid with a head that loves water, but a tail made out of lipids that doesn't like water. So if you throw phospholipids into a solution, they're not very stable. The legs will be kicking, trying to knock the water away. And in the process of using all that energy to knock the water away, the molecule could actually break down. So a phospholipid just stuck in water by itself will not last very long, because it will expend its energy trying to repel the water away from the lipid, which are the legs on this molecule. However, if you put a number of phospholipids in solution, first they come together and create a structure like a ball called a micelle. And that ball, guess what? All the phospholipids are organized so that the heads around the periphery of the ball, the heads are all positive and negatively charged, so they connect with water. So the outside of a micelle can live in the water and be very happy. And the lipid legs that hate the water, guess what? Now they're on the inside of the micelle. So in the illustration, you can see the molecules will assemble to create a structure that is very stable, because inside the lipid legs, there's no water in there. And the outside of the phospholipids, the phosphate groups, are touching the water, so this little group called the micelle is very stable. But as you can see, if I start to add more phospholipids to my micelle, they all can't fit in the circle anymore, and it turns into a bilayer. That means double layer. It's a crystalline structure. Look at it. On one side, all the phosphate groups are on one side with the legs facing down, and on the other side, the phosphate groups are on the bottom with the legs facing up, and we make something that looks like a bread and butter sandwich. The phosphate groups connect to the water, just like in a bread and butter sandwich. The bread can absorb water. But in the middle where the butter is, that's where the lipid legs are, the fat. They resist water. So a membrane is like a bread and butter sandwich, where each side is like the bread, where the phosphate groups are, and they can attach the water on the outside of the cell and water on the inside of the cell. But between the phosphate groups are the lipids, and they're the oil. That's the butter. And I say, well, why is it relevant? So I say, for example, if I add some blue dye to the top of a bread and butter sandwich, pretend the bread and butter sandwich is a piece of membrane, and I add blue dye, let's say that's food, and I drop it on top of the sandwich, and the blue dye soaks into the bread. The question is, did the blue dye go through the bread and butter sandwich so that the blue dye as food could go inside and nourish the cell? So I cut the bread and butter sandwich, and look what you see. when the blue dye hits the butter layer, it stops. It can't go through. A membrane is like a skin. It's a barrier. The lipid in the center doesn't let things in and out. So it separates the inside of the cell from the outside world, so that what's on the inside, I can control what's on the inside. And if the outside is going through all kinds of changes in the environment, inside the cell, I can keep it stable and regulate the activity. I can regulate the pH. I can regulate the salt. I can regulate the temperature. So a skin is a barrier that separates the outside from the proteins on the inside, and the proteins inside are creating the structure of the cell. But the proteins on the inside are regulated by signals. But we have a problem. If the membrane is like that bread and butter sandwich, I can't get food in the cell, and I can't get waste matter outside of the cell. So I say, well, a membrane just made out of phospholipids can make a barrier, but it's not very useful, because if you can't get food inside the cell, then how are you going to create life in there? So I say, there's another component in the membrane, and that are proteins. And there are two different kinds of proteins that are built into the membrane, two different classes of proteins. One class of proteins has antennas on it, just like television antennas. They're called receptors. The other type of proteins are called effectors, and they point into the cell. So receptors reading the outside environment, what's going on in the world? And then what it's going to do is connect to the effector. Many of them are called channels, so let's talk about a channel. What's a channel? It's a protein that is closed, like my fist. And I say, yeah, but what happens when a signal hits a protein? We change the shape. So watch what happens if a signal hits my hand. My hand opens up, and now there's a channel that goes through. So a protein channel built into a membrane, when it's closed, nothing goes through the channel. But when the right signal comes, the protein opens up, and then signals can go into the cell, or signals can come out of the cell through the channel. So a membrane made out of phospholipids is a barrier, a skin. But when you put proteins in there, and we have two different kinds, receptors and channels in this case, I say, what do? The receptor, the name is receiver. And guess what? A human body and a cell are very similar to each other. Why? You have receptors. Where are they? Eyes, ears, nose, taste, touch, pain, temperature, pressure. Where are your receptors located? And the answer, in the skin. I go, it's exactly the same at the molecular level in the membrane. In the membrane, there are protein receptors that act like eyes. They can see light. Some protein receptors see chemicals such as sodium or potassium or calcium or sugar, whatever is out there. And so the point is this. A cell, like you, can see the environment. We have very complex receptor. An eye is very complex. A nose is very complex. But at the level of a cell, the receptors are more simple. They're proteins with antennas on them. And when the antenna sees a signal, it causes the protein to change shape. Stop for a second. I'm a receptor, and I have an antenna looking for a signal. And as a signal shows up, guess what? Then the signal binds to the protein. It causes the receptor to change shape. And when the receptor changes shape, it can connect to the channel. But when the channel is binding to the receptor, it changes shape because it's another protein. The receptor is the signal. When it binds to the channel, it causes the channel to open or to close. And I say, well, what does this mean? The membrane reads the environment. How? The same way your eyes, ears, nose, taste, and touch are reading the environment in your skin, the molecules of proteins called receptors are reading the environment for the cell and sending that information in via the channels, which are opening and closing based on the signal. So all of a sudden it says, the cell can read the world outside. And by opening and closing the channels, it can let some things in the cell, or inside the cell, when the channel opens, things can come out of the cell. And so I can create a flow of information, either information coming in, such as food and nutrients, or information going out, such as waste products. So all of a sudden I say, the membrane has two basic functions right now. One, barrier. Separate the inside contents from the outside. Why? The outside is always changing. The temperature's changing, the daylight and nighttime is changing, the pressures are changing, the chemistry around you is changing. I don't want those changes to affect what's going on inside the cell, because the proteins need a very specific environment to create their behavior. If it's not the right pH, if it doesn't have the right chemistry in there, if it's not the right temperature, the protein won't work right. So the skin of the cell, called the membrane, is like the skin of your body. It keeps out the things we don't want from going in. But the cell can actually read the environment using what? Receptors, like eyes, ears, and nose, but they're proteins with antennas. And as they read the environment, they can recognize what's out there, because if I activate a calcium receptor with what? A calcium atom. A calcium atom comes in, binds to the calcium receptor, the receptor changes shape, causes a channel to open or close and send information to the cell that calcium is out there. Now, if sodium comes, it's not going to connect to the calcium receptor. If there's going to be an influence from sodium, guess what I need? A sodium receptor. If I need to know if there's glucose, is sugar out there, as nutrients, then I have to have a glucose receptor. Simple point. Everything that the cell can see in the environment, it can see because there's a specific protein receptor that can recognize that signal. Again, consequence, signal, when binding to the protein, causes the protein to change shape. So when a receptor reads a signal from the environment, it can change shape and cause another protein in the membrane to do a function. So basically, the membrane of the cell is like our skin. And built into the membrane are these receptors that read the world, and they can relay the signal into the cell using the other class of proteins, which include channels that open and close, and in the process allow things in and out. So, the membrane is reading the world. It reads not only what's going on outside the cell, the membrane has receptors that can read what's going on inside the cell. So the skin is the interface between the outside and the inside. And as a result, the membrane or the skin can read the condition of the world outside and read the condition of the world in the inside. I say, why is it relevant? Because as the environment changes on the outside, the changing environment can be relayed inside the cell, so the cell can make a response to deal with what's going on in the world. Because when the signals from the membrane go inside the cell, what do they activate? The proteins that create the structure and the behavior. So an environmental signal picked up by a receptor sends a signal inside the cell that activates proteins to do their function. Okay? So it's very important to recognize the membrane is more than a barrier. The membrane processes information. Environmental information. What's going on in the environment outside the cell? And the membrane has receptors reading what's going on in the environment inside the cell. And by being able to read both environments at the same time, the cell can adjust the behavior of the functional proteins by the membrane receptors reading the signal, sending a signal into the cell, altering the proteins, and changing the function. So the behavior of the cell is controlled by the proteins built into the membrane, the receptors and the channels. I go, why is it relevant? Because by definition, here is the chemical definition of a cell membrane. The lipid molecules, the phospholipids that make the structure, they're crystalline because they're all lined up. As you saw in the picture, all the molecules are lined up in an order. When molecules are in a structural order, that's a crystal. So the membrane is a crystal. But remember this, when it was just phospholipids alone, nothing could pass through the middle layer, the equivalent of the butter where the lipid was, so no conduction. So a phospholipid membrane is a crystalline structure, but it's a non-conductor. Nothing can get through. But we add proteins. I go, oh, well, the channels in the proteins can let certain things in. A channel can open up and let glucose in. A channel can open up and let potassium in. There's sodium channels that let sodium in. There are chlorine channels that let chlorine in. I say, with channels, certain things can come into the cell and get out of the cell. So all of a sudden I say, it's not a non-conductor anymore. I can conduct certain things. But wait, does it conduct everything? No. The membrane doesn't conduct everything from the environment in and out of the cell. It only lets certain things in. So it's not a non-conductor, but it's not a full conductor. It's a semiconductor. And the proteins that do the conduction are called receptors and channels. So if I give you the definition of the membrane, here's what it is. It's a crystal semiconductor with receptors and channels. Another word for receptor is what? Gate. So let's read the new definition of a cell membrane. It's a crystal semiconductor with gates and channels. It might sound familiar to you, that definition. In fact, the first time I wrote that definition back in 1985, the moment I wrote it, I said, a crystal semiconductor with gates and channels, where did I read that before? And that's when I bought my first Macintosh computer. And I bought a simple book on understanding how your computer works. And guess what? In the first pages of the introduction, there's a definition of a computer chip. What's the definition of a computer chip? A crystal semiconductor with gates and channels. At first I said, oh my goodness, the cell membrane and the computer chip have the same definition. But as I started to look at the function of each of the parts, the lipids, the receptors, the channels, I started to recognize, oh my God, the function of the cell membrane is exactly the function of a computer chip. Well, the computer chip is built out of silicon molecules. The cell's computer chip is based out of carbon molecules. So a cell membrane is a carbon chip. In our conventional technology, the computer chip is a silicon chip. So guess what? A cell has a membrane, which is a chip. It's an information processor. What information is the membrane processing? What's going on in the world on the outside and what's going on in the world inside the cell. And I say, well, in this role, the cell membrane is more than just a piece of like plastic wrap to hold the cell together. It's a primary functioning element of all cells. And that's why all cells, from the most primitive bacterium to the most advanced animal on this structure planet, have the same structure of the membrane. A bacterium and a human cell have the same membrane structure. It's fundamental to all of life. That membrane structure functions as a computer chip. I say, what does it do? It reads the environment and then sends information into the cell so that the cell can adapt to whatever's going on in the environment. So I'll give you a simple example. You walk outside and it's hot out. Your skin feels the temperature, and the hot temperature picked up by the skin causes the body to change its function. It causes us to perspire and cool off. But if you walk outside and it's cold out, when the skin picks up the temperature of cold, it changes the function of the body, but it doesn't cause it to cool off. It causes it to warm up. It increases the metabolism. So guess what? The temperature of the body is not controlled by genes. It's controlled by the environmental signals. And this is the way cells work. The functions inside a cell are not controlled by genes. They're controlled by the signals from the environment. Signals plus proteins create behavior. So all of a sudden start to see is, what is the actual role of a membrane? Well, number one, it's the nervous system of the cell. It is what is reading the world, stimulus, and then making a response. The nervous system is stimulus response. That's what the membrane does. And I say, in creating this stimulus response, what functions are associated with it? Well, number one, it keeps balance inside the cell. Homeostasis, harmony. As the temperature on the outside warms up, what it does is it causes the temperature on the inside of the cell to cool off. If the temperature outside is cold, then the function of the cell membrane is to adjust the metabolism so the cell stays warm. What's the point? Proteins operate at a very specific temperature range. If it's too cold, they don't operate. If it's too warm, they don't operate. So what's the function of the cell membrane? To read the temperature and adjust the body. It's called homeostasis. That keeps us in balance. And if the chemistry on the outside changes, the cell membrane can change the chemistry inside. Again, homeostasis to keep it in balance. But then the membrane is information that adjusts the functions of the cell. It adjusts the respiration of the cell, the digestion of the cell, the movement of the cell. So the membrane is creating self-organization. It is organizing the function of the cell to stay in harmony to what's going on in the world. If there's a predator coming, the cell can read the predator coming. It's going to adjust the movement of the cell to move away from the predator. So it organizes the function inside the cell. In addition, the channels opening and closing let nutrients into the cell that we can use for our metabolism. And the channels can also get rid of the waste matter so we don't become toxic inside the cell. So the membrane now, of course, is involved with metabolism. And then it has to read the environment to see, should it grow or should it protect itself? That's two different functions. Well, if the environment is supportive, then what's the cell membrane going to do? Select the elements from the environment, bring them into the cell membrane via the channels, and once those elements are inside the cell, it can control the growth of that cell. As I said, it also is really a continuous process of adaptation. By reading what's going on the outside of the cell, the membrane can adjust all the activities on the inside to stay alive. So if the environment changes, the biology on the inside can change to stay alive and complement the environment. That's an adaptation. And of course, what's the function of the receptor and the channel combination? The equivalent of the nervous system. The receptors respond to the stimuli, the environmental signals, by creating a behavior when they connect to the channel, which changes the function inside. So a stimulus from the environment leads to a response inside the cell. And then, when it comes to reproduction, the cell membrane is also involved. Because when I have to separate a cell into two cells, I have to cause the membrane to come together, constrict, and separate cell 1 from cell 2. If the cell membrane can't separate the two products, then the cell would never be able to divide, reproduce. So when you look at all the functions the cell membrane is carrying out, originally biologists thought it was just a skin, a barrier to keep out unwanted things and protect the inside. But what do we now know? Well, the membrane is involved with homeostasis, balance between the inside and the outside. It's involved with self-organization because it will read the environment and organize the function of the cell to carry out those behaviors necessary to stay alive. It's involved with metabolism. Yes, the membrane is the barrier that separates the inside from the outside. So if you want to bring in nutrients to metabolize, it's the role of the membrane to control that. It controls growth processes by allowing information to come in to adjust the dynamics and the behavior of the system and allow the system to reproduce itself. It's also, as basically, environmental adaptation. Why? Well, if the membrane can read the environment and tell the cell what to do, as the environment changes, the cell can adapt. That's as simple as that. It responds to the stimuli outside like a nervous system does. The nervous system, eyes, ears, nose, taste, what are they doing? Reading the environment so I can adjust my biology, stimulus response. Same role for the membrane. And if you want to reproduce a cell, it has to divide. That means you have to take the membrane that was holding one cell, pinch it off, and make two cells. If the membrane is not involved, you can't separate the cell into two different ones. So the membrane is a fundamental part of reproduction. So the membrane is carrying on a profound number of functions simultaneously. And in fact, in very primitive organisms like a bacterium, there are no organelles inside that carry out different functions like mitochondria, nucleus, and all that. All of the functions are controlled by the cell membrane. The life of a bacterium is expressed through the behavior of the cell membrane: growth, metabolism, responding to the environment, reproduction. All of these are because the only real structure in a bacterium, in all bacteria, is the cell membrane. So, interesting point. Until recently, biologists didn't think the cell membrane was any more than just a barrier to protect the inside from the outside. But in the last 30 years, we're beginning to find that the membrane is the most important organelle in the cell. Organelle, that means miniature organ. There's organ, and then organelle means miniature organ. So in a primitive bacterium, the only organelle structure is the cell membrane. But as you get into bigger kinds of cells, something like the amoeba, then we find, oh my goodness, there are all kinds of organelles inside there. Used to be the function of the outer cell membrane in a primitive cell, but in more advanced cells, the membrane functions were taken into the cell and created membrane organelles that did things like respiration, digestion, excretion, contraction, and mobility. So primitive cells called prokaryotes, bacteria, don't have a variety of organelles in them. All of the functions are actually controlled by the cell membrane. But in larger cells that have a nucleus, and these are called eukaryotic cells, there are also other structures in there that are carrying out functions that once were done by the cell membrane. But in higher organisms, those functions are taken off the surface, and using membranes on the inside are controlling those functions. So the membrane used to be considered nothing more than like plastic wrap to hold the contents together, and now we find that almost all of the functions are mediated and controlled by the cell membrane. The cell membrane is the equivalent of a chip. It processes environmental information, and in that processing then controls the contents inside the cell to carry out behaviors to keep that cell alive. So, interesting point. What was once a useless organ, skin, becomes one of the most important. And guess what? In a human, the nervous system during embryology comes from the skin of the embryo. It's just like the cell. The skin of the cell is the cell's nervous system. Massive functions, very important to study. In our story of evolution, we talked about the fact that before cells formed, they were actually precursors that are called biofilms. And biofilms were membrane-trapped group of bacteria. And the bacteria carried out different functions. They weren't all the same bacteria. Some bacteria could breathe oxygen and make energy from that. Other bacteria were called anaerobic. They actually could live in an environment with no oxygen. And some of the bacteria were creating certain proteins, and other bacteria were involved with respiration or removing the waste from the community of the biofilm. So the original precursor to a cell that we talk about, such as an amoeba cell, a eukaryote, was a community of different bacteria, up to hundreds of bacteria, living in the same membrane. Well, it wasn't efficient to have that many different living organisms under the cell membrane. So over time, every different bacterium was different because of what? It had different DNA in it, and it had different programs in it, and it had different behaviors. And yet, to live together in a community, different bacteria had to contribute their functions to the rest of the bacteria as well. So they were sharing functions as individual bacteria. So there were bacteria doing respiration, some doing digestion, excretion, others being a nervous system, for example. I say, well, the efficiency of so many cells, bacterial cells, prokaryotes living under the membrane, was not that efficient. And nature saw that, look, why do I need the whole bacterium? Because it's the nucleic acid, the genes in the bacteria that provide for the function. So instead of having a whole bunch of different bacteria, what would happen is the DNA from the individual bacteria started to be collected into one big structure called the nucleus. And the point about it is this: the nucleus has all the genetic information to do all the different functions that hundreds of bacteria were doing. But guess what? I don't need hundreds of bacteria now, because the nucleus can just make the proteins that were necessary to do the functions. So I say, yeah, in making these proteins, the cell had to create structures inside the cell that were doing the same function as the different bacteria. Most of these structures have membranes involved because, remember, the function of bacteria is almost all in the membrane. So if I'm going to take a function from a bacteria, I'm going to have to use it in a membrane because that's the way it worked originally. So inside the cell, there are different kinds of membrane structures. And these structures carry out the functions of the systems that are present in a body. So, as mentioned, reproductive, respiratory, digestive system, nervous system, even musculoskeletal system. These are functional groupings that in the human body, we give these functions to different organs. So the lungs are part of the respiration, the stomach, the rest of the gut is digestion, the kidneys are excretion, the muscles and the bones are the structure called the musculoskeletal system. And I say, all of these functions were already present in the most primitive cell, called a eukaryotic type cell. That's the one with the nucleus. That's the one with all the genes from the different bacteria. Why? Go back one step, and you recognize in the prokaryote world, individual bacteria can carry out individual functions because they have very specific genes for each function. But when the bacteria came together and they started sharing, then the significant evolution was take the DNA that made each bacterium different and put all that DNA into one structure, one nucleus, so that one single structure in the cell can program all the behaviors that represent the systems. But when they created these systems, they weren't organs, giant organs like we have. They were small single cells. And so I say, yeah, but all the functions of a human are already present in the single cell. Yes, you show me a function in a human body and I'll show you in the cell where it is. Reproduction, the nucleus, that's where all the DNA is, okay? Brain, oh, that's the cell membrane, okay? Respiration, the lungs, well, in a cell, that's called the mitochondria. The synthesis that we find in the digestive system to make all the enzymes like our pancreas and our liver, well, those are the functions of membrane structures called endoplasmic reticulum and the Golgi body. So what am I saying? I'm saying all the functions of our organs in a human body are already present in the single cell. And they're the equivalent of organs. They're not the same of organs. They're actually very miniature organs in a cell. And the name of these miniature organs is organelles. Organelles. The L's part represents miniature. So organelle means miniature organ. So I say the structures inside a eukaryotic cell, like an amoeba or a human cell, these structures that are in the cytoplasm, which makes up the bulk of the material underneath the membrane, these structures are miniature organs. They're called organelles, and the organelle functions are the same as the organs in the human body. So it's an important point to mention right here, right now, and that is this. A human is a community of amoebas. And I say, well, what about it? I say, well, some amoebas are muscles, some amoebas are brain, some amoebas are skin. And all of a sudden you see the amoebas take on different functions in our body, but collectively they all work in harmony to create one human organism. The important message here is very important, and that is this. There's not a new function in the human being with 50 trillion cells. That's not already present in almost all of your cells. Almost every cell in your body has every system that you have. Cells have a respiratory, digestive, excretory, nervous system, endocrine system, reproductive system, musculoskeletal system. Cells even have an immune system. The point is this. All the functions of a human being were already programmed into the structure of the single cell. And so a human is an enlarged version of a cell. We're carrying out all the functions that cells can do because the only way we can do anything is because we have cells that are doing it. So if we have a digestive system, cells have a digestive system. And in fact, the protein enzymes in the cell digestive system, such as in an amoeba, are the same enzymes that the human uses to digest its food. So there's an important connection. All the functions of the human are already present in most all of our cells. And so the interesting part is this: if you want to understand how a human works, you don't have to study the whole human. You could just study the single cell, because the mechanisms of life that we express as humans are already present in those single cells. And these structures inside the cell, miniature organs or organelles, and this is, when we look inside the cell, this is what we find. So what we have are, in the very beginning, before the evolution of the amoeba, is called the biofilm. We have lots of bacteria living together. Well, they weren't just in a soup. The bacteria started to make fibers and filaments and things called microtubules, like skeletal structural elements. So if you can imagine in your head, if you went inside a cell, the first thing you would see is a structure that looks like a sponge. It's got all the walls around it, but a sponge has got holes in it and channels and connection. And guess what? It's in those channels and connections that the bacteria live. And so all of a sudden I say, when you open up a cell, there's actually a sponge-like structure made out of microfilaments called actin, intermediate filaments, microtubules, and these are the equivalent of a cytoskeleton. And the relevance about all this is really important, for the main reason is this. Inside a cell, there's already structure like a sponge. But then, remember, the bacteria were living in the channels of that sponge. But when the evolution occurred and the bacteria just left their DNA and their bodies were no longer present, then the DNA created the functions called organelles. And the basic ones, as mentioned, mitochondria, nucleus, endoplasmic reticulum, Golgi. These are the names of organelles. And guess what? They're inside the spaces inside the sponge. And so the relevance is very simple. A cell is like a sponge with filaments and fibers, which are like the musculoskeletal system. And between all the filaments and fibers are spaces and holes where the organelles are doing digestion, respiration, and excretion. So inside a cell, it's not just a sack of membranes in there. There's a structure. And that structure is very important. That sponge-like structure is important because you know what it can do? It can contract and it can expand. And that's how a cell, like an amoeba, creates what are called pseudopods, fake feet, fake legs. The pseudopods are extensions of the cytoplasm that stick out. So an arm in a human is like a pseudopod in an amoeba. I can stick out the arm, but it's a permanent structure. Amoeba, they can stick out an arm and then pull it back in, stick out another arm over here, pull it back in, stick out an arm, and these are called pseudopods. And the movement and the structure of the cell is due to the extracellular matrix. What that means is this: there's a matrix that's not the cells, and that this matrix forms the sponge. And the sponge has the channels in it where the organelles are carrying out the function. So there's an anatomy inside a cell. It's not just a bag of membranes, a complete structure. And so there are organelles and membrane structures and filament structures that provide an anatomy for a cell. It is not just a collection of proteins inside a membrane. There is a complete understanding in a eukaryotic cell that every function that is present is being carried out by a miniature organ, an organelle. and that these organelles are integrated into the cytoskeleton, the matrix that gives the cell its basic structure and its ability to move. So it's important to recognize that a cell is a highly organized structure and has an anatomy that is very complex, but our anatomy is a reflection of the same thing. Instead of actin filaments and intermediate filaments and microtubules, what do we have? Muscle filaments. We have bones, which are like the microtubules. So the functions in a human are already present in the eukaryotic cell. Wherever there's a function of an organ in my body, there's an organelle in that single cell that's doing that same function. And this is a beautiful understanding because it allows scientists to not study the whole human to figure out how it works, but more simply, study the role and function and structure of the cell, because the information we get from that research applies directly to understanding the structure and function of the human body. And therefore, life is highly organized from the very primitive cell to the most advanced organism on this planet. It's not random. There's a complete structure that is repeated from the cell level to the human level.
Hi dear friends, cultural creatives and imaginal selves everywhere, Bruce back. And this month's newsletter I really want to focus on something called an urban myth. And this particular urban myth is the belief that we only operate off of 10% of our brain. So we've all heard that yes, we're only using 10% of our brain, and boy, if we could use all the rest of it, we'd be super smart. But hey, that's all we use. Firstly, where did that number come from, 10%? A long time ago, when putting the body together through an understanding of histology and the cells that make up organs, a study of the brain revealed one very important fact, and that is only 10% of the brain was made up of neurons. 90% of the cells in the brain were not neurons, but they were called supporting or connective tissue and given the name glial cells. Well, what's really interesting about that is in the neuron principle, the neuron doctrine, it was believed that the neurons were the functional cells of the brain and the glial cells were more or less just support. Well, then by definition, right away, it says then you're only using 10% of your brain because that's all that the neurons comprise in the brain, 10%. For a long time, the glial cells, again, were thought to be just like connective tissue support physically and nutritionally the neurons. But over the last number of decades, what we started to find is this: there was a misunderstanding about glial cells. That glial cells are a lot more important in neural function than just physical support. Glial cells can inhibit or activate neurons. Glial cells connect to neurons. Glial cells can control the activity of neurons. So all of a sudden, these glial cells, which were looked at as just passive cells in the brain, turn out to be actually quite functional in integrating the nature of how the brain works. In a way of looking at it, in a simple analogy, consider that the neurons are nouns in a sentence, and that the glial cells are the verbs and the adjectives and the adverbs, the modifiers of all the nouns. So the glial cells create a fulfillment of a picture. So neurons are like a stick figure, and when you add the glial cells, you fill out the nature of the whole human. So glial cells become a very functional, integrated part of all neuronal activity. So the concept of the neuron doctrine that said only neurons are involved with functioning in the nervous system, it turns out the glial cells are as well. So now we're not dealing with 10% of the brain, we're dealing with 100% of the brain. Yes, you have access to 100% of your brain all of the time. But does that mean that we actually use the whole brain all of the time? And the answer is no. Because what we start to find out is when we start to live in patterns and in habits, that what happens is we just activate certain neuronal pathways that are habitual pathways. And as a result, these are the pathways that have preference in our everyday life. But if you can engage whole brain activity, you can enhance your consciousness and your awareness and your ability to control your life. So basically it says that we can become whole brain when we start to think holistically instead of just playing recurrent programs that play over and over again and activate the same pathways. So the relevance is that once we start to engage in whole brain activity, we enhance our ability as humans on this planet. And now we're beginning to find out there are many different ways to engage and activate the whole brain. Some of them involve simple exercises such as something called brain gym. And brain gym is a very interesting exercise of the body and the nervous system integration in this way. My right hand is controlled by my left hemisphere. My left hand is controlled by my right hemisphere. But here's an interesting story. If my right hand, controlled by my left hemisphere, crosses the midline, then it simultaneously gets picked up by the other hemisphere because now it's in that territory as well. So when you cross your arms and when you cross your legs, what you're doing is causing the right and the left hemispheres to work together in harmony. When we are activating brain synchronization, where right and left hemispheres are engaged together simultaneously, it activates something like superlearning. With both hemispheres working together, we have much more superior characteristics of neural function. However, after age 7 and 8, we start to express what is called brain dominance, meaning during the day, we don't have synchronization of both hemispheres. During the day, it's like a wave form. Sometimes we're more in the left hemisphere, sometimes we're more in the right, then it comes back to the left and back to the right, so that we're cycling through one hemisphere at a time. This is called brain dominance, where one hemisphere is dominant over the other hemisphere. Well, the relevance about that is integrating our lives is very difficult in brain dominance for a simple reason. As been suggested, our left hemisphere is based on logic and details. Our right hemisphere is actually associated with emotions and wholeness. And this becomes very important because if you're only looking with your left hemisphere, then all the logic things make sense, but there's no emotional component to it. And reverse, if you're in the right hemisphere, you get emotionally tied up, but the logic drops out. Well, this happens to us ever since the time we were 8 through our adult life. If you can get both hemispheres to be in sync, hemisync, then you're engaging logic and emotions at the same time. That opens up a window for super learning opportunities where you can download information very, very quickly and very fast, but it also opens up a whole wide range of thinking that includes an emotional component as well as an intellectual component, which is necessary to fulfill a whole picture. So if you're operating from brain dominance, you're actually shutting off one component of your nervous system at that time. So if you do an exercise like brain gym where you're involved with crossing your arms and your legs, when you do that, you start to integrate both. Very interestingly, when we have hemisync, we're much more calm, collected, and able to really express neurological functions that are very fully supporting of ourselves. And this is why it's very interesting when you go home and relax at night, sometimes you find yourself with your ankles crossed over each other just sitting in your chair. Without even knowing what you're doing, by crossing your ankles, you're actually engaging a whole brain process. So if you're stressed, for example, during the day, or you just want to relax when you go home at night, crossing your arms and legs actually engages both hemispheres, which calms the system down, but engages a whole brain process. So you become much more effective in your relaxed state. So if you're having a tough moment during the day, just take a moment and cross your arms and legs. Hold them together and just wait for about 3 or 4 minutes and you'll start to feel a calmness overcome. And in this calmness, your neurological functions will be totally enhanced. When we start to operate from patterns that are replayed every day, we start to express very specific pathways that are reused over and over and over again. And as a result, we actually shut down a large amount of functioning of the brain and just go through the habit, the patterns that replay themselves over and over again. So basically, what you really have to understand is this: we can use 100% of the brain, but if we play the same patterns over and over again, we don't need to use 100% of the brain. So, simple point, I'd just like to suggest that once you start to recognize that you can have hemisync, where you can get right and left hemispheres to engage together and stay conscious, you are using 100% of your brain activity. And when you do that, you'll also find that your life is easier, more harmonious, and healthier. Because when you're using your full nervous system, it will support your life and enhance your growth. And this becomes important. So don't get stuck that I'm only using 10% of my brain. That story is false. You can use 100%, and it's available to us. And basically all you have to understand is this: stay conscious, stay mindful, and stop playing the same patterns over and over again, because that opens up an opportunity to create new behavior when you stop playing the programs. Hi, dear friends, cultural creatives, and seekers everywhere. Bruce here with a little suggestion about the nature of life and fitting in or not fitting in, conforming or not conforming. And why am I talking to you about it? Well, right away, I can tell you, if you're watching this show, you're not necessarily conforming to the world, because you're the cultural creatives. You are the people that are looking for new answers and new insights to understand how we can have a better and healthier and happier life on this planet. And I applaud you for that. Energy in the field can do either of two things. Energy in the field could be motive force. Yes, I need the energy to move and use this to push and do all that stuff, motive force. But there's a lot of energy in this room right now in which I'm sitting has no motive force at all. I'm sitting in an aquarium of energy. There's cell phone broadcasts running through here. There's television broadcasts, radio broadcasts. Solar energy is passing through this building as well. And I'm in this aquarium of all this energy. And I say, yeah, but this is not motive energy. This is informational energy. If you take iodine tablets, then you're providing an iodine base to your biology. Simple point. If you flood your biology with excessive iodine more than you need, then the biology will not necessarily start scavenging the iodine from the environment. And even if you pick up some iodine in the environment in synthesizing the thyroid hormone, you'll have so much abundance of natural healthy iodine compared to the invasive radioactive iodine that the system will preferentially just use the good iodine just because it's competition. So, the biggest fear we have is actually incorporation of iodine, affecting the thyroid hormone, which really totally influences the entire metabolism of our body. So therefore, radioactive thyroid hormone is obviously not a good thing. You follow the path of your heart, and you will come to learn, as I did, that we are living heaven on earth. And that heaven is not found in the box. It's found outside the box. Free yourself, free your mind, take that red pill. Go for the experience. Live with your heart. That brain is really great. I know that. I lived with it for a long time. But it's not as true and pure in its conclusion as listening to the voice in your heart.